and how this loop of Henle acts as a countercurrent structure, right? So we have a descending limb and it makes a hairpin turn, and a ascending limb that comes up. And as you follow the ascending limb up, notice it's thin in width for, a third of the way up, at least in juxtamedullary nephrons. And then most of the ascending limb is thick of structure. It's got a thick epithelium. And the reason for that is because this epithelium of the thick limb is going to have a lot of mitochondria in it. It's going to actually pump sodium chloride into the extracellular space. And so instead of being a classical exchanger, it's actually a countercurrent multiplier. So part of it exchanges or uses diffusion to move sodium chloride into the medullary interstitial fluids. And the top portion of the ascending limb is going to be a multiplier because it's actually going to expend a ton of energy pumping sodium chloride into the extracellular space of the medulla. And the medulla is going to become very hypertonic to the blood supply. And what that's going to allow is the collecting duct, which the nephron is connected to kind of tweak the final volume of urine that is going to be produced by individuals, depending on your hydration, right? Whether you're hydrated or not very hydrated. And it's going to be under hormonal control. And probably one of my favorite things to do, as you will see later when I show you slides that'll freak you out, is understand the mechanisms, right? The mechanisms are really cool, how these epithelia interact with hormones, and how the distal convoluted tubule plays a huge role in sensing what's going on within the nephron. And so we're going to start to get in and do that here in a few minutes. So remember, the descending limb is permeable to sodium chloride, it's permeable to water only. And so that's why your numbers go up from 300, 400, 600, 800 to 1400, right? So you're concentrating the filtrate by losing some water. What drives the water out is that hypertonic environment. By concentrating this, we can then take advantage of at least part of the ascending limb for just simple diffusion, right? Because we got such a concentrated amount of sodium chloride. If I become permeable to sodium chloride as I ascend, then it will leave to the filtrate into the interstitial space until it reaches close to equilibrium, right? Once you hit that point, then the filtrate is entering the thick part of the loop of Henle, and you can now pump sodium chloride against its concentration gradient, maintaining very hypertonic conditions within the medulla. And this is all allowed or is effective because of the countercurrent multiplier. So here's how we move those ions. So this is the ascending limb, and that's the one I'll pick, that is utilizing sodium-potassium pumps, and it's using lots of sodium-potassium pumps. The epithelium is a very tall cuboidal epithelium, so almost columnar, but it's a pretty thick cuboidal epithelium. And the basal compartment is filled with mitochondria. All right, and those mitochondria are driving these pumps, and the pumps are going to be very common in the basal membrane of the cuboidal epithelium of the ascending loop. And we are going to be pumping sodium chloride, keeping sodium concentrations really low inside of the cells that line the thick ascending limb. And we're going to use a symporter here. And remember, these symporters, this is the sodium chloride potassium symporter. And they have a name. Let's see, hopefully this marker works that's on the floor. Yeah, they're called NCC, so sodium chloride potassium, right? NCKC transporters. So you got sodium chloride potassium co-transporter, and you're using the sodium gradient. And this actually, there are a couple different types, and the one that's found in the kidney is type 2. I don't really care that much about that. But the kidney-specific sodium chloride potassium co-transporter is found in kidneys only, which is cool because this variation in the amino acid sequence of this protein allows you to produce pharmaceutical drugs that are specific to the kidneys. As a matter of fact, they use what are called loop diuretics here. If you ever have to take a diuretic because of blood pressure or because of edema like you talked about, some of the first order loop diuretics, which means to produce more urine, will block these sodium chloride and potassium exchangers, which will increase urine volume, all right, the amount of urine you produce. And we could talk about why after we get done with this, so see if you guys understand what's going on here. But we're bringing sodium in, right? And we are going to dump it into the blood supply and eventually into the interstitial space of the medulla. Now notice here how chloride travels. Chloride actually travels transcellularly in the loop of Henle, which is completely different than how it traveled in the in the proximal convoluted tubule. In the PCT, remember the chloride traveled paracellularly, remember? It followed sodium through a paracellular chloride channel. Here we're using this co-transporter to bring chloride and potassium in along with sodium, pumping the sodium and allowing the chloride to follow the sodium into the medullary space. Notice we have 2 sodium pumps here, by the way, also, which I should point out. We not only have a primary active transport pump that uses ATP directly, but we also have a secondary active transport pump that's a symporter here using bicarbonate as our driving force that pushes sodium into the blood against its concentration gradient. And we know we can get bicarbonate from carbon dioxide, right? By converting carbon dioxide into carbonic acid and then breaking that down into bicarbonate and hydrogens. And this is how we get rid of acids into the filtrate. And notice that helps. We pump those hydrogens out using sodium, right? So this is a secondary active transporter using sodium as our driver here that drives protons out. That's an antiporter. And this allows us to control our acid base, or our pH of the blood supply also. But this is how we move all of that sodium and chloride from the filtrate into the medullary space, and that will dilute, right? That will dilute our filtrate as it comes up. So if I back up a slide, that's why our numbers are going up, right? And when you hit 600 here, then we get into the ascending limb and we really pump the sodium out in that thick portion of the ascending limb so that at the top of it you're at about 100 milliosmoles. So you're actually hypotonic to the blood, right? As far as solute concentration. Now notice as we pass through the distal convoluted tubule, we're going to continue to move some water and some solute, but we're going to move it equally. So notice if you're around 100 here, or whatever number you enter into the DCT, you're going to end the same way. You're just going to have less filtrate, right? So by the time that I'm going to dump this filtrate into the collecting duct, over 80 or 85% of the entire filtrate has been reabsorbed, right? So you're left over with just a small portion of this primary urine, which is going to become definitive urine. And now the collecting duct is going to tweak how much of that volume I'm going to allow to escape. And it's really going to be water, right? How much water am I going to allow to escape? And it's going to be hormones that are going to communicate that information, right, to the collecting duct. Does the countercurrent exchanger make sense, how it works, and why it's a multiplier, and why that would cause a hypertonic environment? So if you go through all this, and if you really can understand it, I would draw it, talk your way through it. Then I want you to go back and I want you to think about this. I want you to think, okay, if I use a drug, a loop diuretic, and I block this co-transporter, why would that lessen my ability to concentrate urine, all right, or to produce less urine? So if you're taking a drug that blocks this particular co-transporter, that's really going to affect your ability to produce concentrated urine, okay? So you're going to be peeing a lot. You're going to get rid of a lot of water, right? You're going to dump a lot of water. So this is, for example, if you have slightly high blood pressure, right? The first kind of drug they like to start with is typically a loop diuretic because it'll drop blood volume and that'll drop your blood pressure, right? A little bit. You got to be careful with it because it also affects potassium concentrations, and potassium is a very important ion as far as nervous and muscle contraction. But you know, a low dose loop diuretic often helps people. If that doesn't help you, then they'll go into using what's called a beta blocker, and we'll talk about how those work when we get to the cardiovascular center. So go through this, make sure you understand the importance of this co-transporter, and if you think you understand how this whole process works and how it causes this hypertonic environment in the medulla, if I block this, it's going to affect urine production, okay? All right, so let's talk about the DCT and the collecting duct. Now, the one thing we're going to get into here in a second is notice as the DCT comes up, it is going to be brought up right next to where the afferent arteriole enters the renal corpuscle. That is on purpose, all right? And the reason for that is because the epithelium right here that lines the DCT is going to enlarge. It's going to go from a cuboidal to a tall columnar group of cells. And those columnar group of cells are going to have a special name. They are going to be called the macula densa. All right, and the macula densa is a sensing epithelium. All right, and it's going to sense a number of control conditions to basically make sure flow rate of the urine is consistent. And we're going to talk about how it does that in a few minutes, hopefully. So once the filtrate reaches the collecting duct from the DCT, about 80 to 85% of the water has been reabsorbed, along with many of the solutes, right? So what you're left over with are waste products and some water. Now, the DCT and the collecting duct, they continue to reabsorb sodium chloride, urea, and bicarbonate. So if you haven't noticed, these are really important molecules, right? Or ions. One's a molecule, or two are molecules and two are ions. Obviously, the most common extracellular ions in your body are sodium and chloride, right? And that's on purpose because the kidneys reabsorb this all along the nephron, and those play very major roles in those electrochemical gradients that we've talked about so much that drive transport and eventually drive nerve and muscle contraction, as you'll see. We know that the interstitial fluid of the medulla is very hyperosmotic, right? Because of our countercurrent multiplier. So now what we can do is we can use that environment to drive water volume. All right, and so how do we do that? Well, it's pretty simple. The collecting duct can be either permeable or not permeable to water. Okay? And if the collecting duct is not permeable to water, then you're going to produce a very dilute urine. So if you're drinking tons of water, all right, good for you. You should be drinking tons of water. Look, I'm drinking water today. Then you're going to produce a very dilute urine, right? The more water you drink, the more clear it is. I mean, the more watery it is, right? So that happens when you get to the point in the above example where we've got to the end of the DCT, right? And I have 100 milliosmoles of volume of fluid, and I'm simply going to allow that fluid to escape, right? So I'm not going to be permeable to water. But along the collecting duct, I'm still losing some of these ions from the filtrate. So notice my number continues to drop, doesn't it? 100, 95, 90, 80, 70, to about 65 milliosmoles, which is about 4 times less than the osmolarity of the blood. So I can produce urine that's 4 times as dilute as blood osmolarity. And this is what happens when you are really hydrated. Or if I'm the opposite and I don't drink water and I'm very dehydrated, then I can produce a urine that this number will go up. It'll go from 100 to 300 all the way up to as high as 1,200 milliosmoles, which is 4 times the hypertonicity of the blood supply. How do I do that? I simply make the collecting duct permeable to water, and water leaves, right? Why does water leave? Because the medulla is very hypertonic, right? And so all I have to do is insert what into the epithelium if I want water to move? A water channel, right? Aquaporins. And that's exactly what happens here. Now, how does the collecting duct know, right? I use the quote unquote know to allow water to escape or to reabsorb water. That's the job of hormones. All right, hormones are going to communicate with the collecting duct and tell it what to do. The main hormone here is ADH, and this is why we're not doing lab today, because I wanted to do this mechanism first before we inject frogs so you kind of understand how ADH works. And what ADH is called anti-diuretic urine, I don't know why I said that, anti-diuretic hormone. So remember, I just told you that diuretic means to produce more urine. So anti-diuretic means what? To produce less urine, right? So ADH is also called vasopressin, right? And so that's what the hormone name is called, vasopressin. What did I do with that? Oh, I put it in my pocket. So vasopressin means it has an effect on blood volume. All right, so vasopressin is another name that you will see in the lab handout when we do the lab. We will be injecting vasopressin, or ADH, into frogs and watching them gain weight. And we'll talk about why and how here. Now, before I move to the next slide and talk about the mechanism of ADH, I like this figure here also because it shows you all of the major movement of solute, if you will. PCT versus loop of Henle versus DCT and collecting duct. And so that's kind of a summary figure, if you will, of all of the things that move along the nephron. So how does ADH work? So ADH is released from the pituitary gland. You guys all know where the pituitary gland is? It's an outgrowth of the brain, right? It comes off the brain. And it's associated with a part of the brain that's called the hypothalamus, right? The hypothalamus, which is located at the floor of the third ventricle, which is one of the major cavities within the brain. And it basically grows out of or off of the brain, and it has 2 lobes. There are 2 lobes to the pituitary gland. There's an anterior lobe and a posterior lobe. The posterior lobe, the back part, or the dorsal part, the part facing the back, your back, is a direct outgrowth of the hypothalamus. That is, it's nervous tissue. All right, and basically it's a bunch of axons. Remember, I've drawn a nerve for you, right? It's a bunch of axons, and the cell bodies of those axons start up here in the hypothalamus, and they send their axon down where it ends in the posterior pituitary. And it produces two hormones, the posterior pituitary, vasopressin and another one called vasotocin. All right, they sound very similar. Vasotocin causes smooth muscle contraction. Vasopressin causes effects on the kidney and decreases urine production. So, the nice thing about the hypothalamus, and the reason the pituitary grows off of the hypothalamus, is because the hypothalamus is a major region of the brain that is participating in maintaining homeostasis in your body. Things like osmolarity, solute concentration, or volume of your body fluids. Temperature is another area that is controlled by the hypothalamus. Right now, your hypothalamus is going insane because it's freaking freezing in here, right? And I'm shivering and I'm lecturing, right? And so you're cold, right? And so the hypothalamus is releasing or controlling a lot of those things that are trying to increase our body temperature. But the posterior pituitary here, when the hypothalamus senses some changes in the blood supply, it can then release this hormone. So for example, the hypothalamus, which is in direct contact with the blood, can sense a drop in blood volume, okay, or an increase in osmolarity. Isn't that saying the same thing for the most part? If you drop blood volume or you lose water, then your osmolarity is going to go up. That is, you have more solute than water, right? And your hypothalamus can sense that because those nerves, the sensory nerves that are the nerves associated with the hypothalamus, are in direct contact with the blood supply and can sense an increase in osmolarity. They're basically monitoring osmolarity all day long, right? That's their job. And when you get an increase in blood osmolarity, that is going to stimulate these nerves responsible for controlling osmolarity, and they will release ADH. ADH will then enter the bloodstream and travel from the brain's blood supply all the way down to the kidneys and will bind to its target tissue. Now, when you hear target tissue, those are the tissues that actually express the proper receptor for our hormone. And so the epithelium that lines the collecting duct and the DCT actually have ADH receptors. And they can bind ADH, and that will communicate with the epithelium to increase water resorption, which helps prevent osmolarity increases or reverses our stress, right? This is a positive feedback mechanism, isn't it? So how does it do that? Well, it's pretty simple. It's going to cause the insertion of water channels. All right, and those water channels inserted into the epithelium will allow water to escape the filtrate so that everything that you are pushing through the nephron, you're going to reabsorb as much water as possible to help maintain water volume. The other thing ADH causes is thirst. Right? You're thirsty. You start drinking. So today I'm very thirsty because I've been drinking too much damn Coke. And now I need to drink water, right? I know, I'm double fisting, right? Now, once we return our blood osmolarity back to normal, 300 milliosmoles, this whole thing shuts off and I stop releasing ADH, right? Now, here's a problem though. If I'm just reabsorbing water, I could actually start to dilute out my solutes, right? That is, if I had too much water, then now I have more water than solute, and that could affect overall body volume osmolarity. And so we are also going to use another hormone called aldosterone, which will help with solute concentration. It also causes reabsorption of water, but with solutes, right? ADH is simply water movements, all right? So it's pure water you're moving. Aldosterone is going to be solute and water, all right? So they kind of work together, as you're going to see. So that's what this last statement here says. To keep my sodium chloride levels appropriate and not just absorb pure water, another hormone will help ADH, and it's called renin, all right, or renin. I've always heard it as renin. I've heard other profs call it renin. You can pronounce it however you want. All right, so we'll talk about this hormone here in a few minutes. So this is cool right here. So this is a study that was done on aquaporins and ADH. So increasing aquaporin in the apical cell membranes of collecting duct cells. Notice the numbers here, all right? None up to 1.6. And notice pre-ADH, you have very few aquaporins. During ADH increasing within the blood supply, you have lots of aquaporins. And then post-ADH, you have just slightly more than pre. So when ADH is present, aquaporin numbers increase in the apical membrane. And just to show that that parallels water resorption, notice permeability here of the membrane also increases with ADH. It makes sense, right? You add more aquaporins, more water's moving. This will concentrate the filtrate dramatically as it passes through the collecting ducts. So the question is, how does ADH do this, right? And I know you all can't wait to learn how ADH does this. And here is your cellular mechanism. So this is an epithelial cell, obviously, right? This is the basal side right here. This is the apical side. Notice our little microvilli. So this is the filtrate over here. This is the collecting duct lumen, and this is the blood supply on this side, right? So ADH travels through the blood from the pituitary gland, right, in response to an increase in osmolarity. And so when it gets to a collecting duct epithelial cell, the receptors are obviously expressed on the basal side, right, where the blood supply is. And they're called V2 receptors or vasopressin receptors, right? V2, there are several types of vasopressor receptors, V2 being one of them. It's a G-coupled adenylate cyclase receptor. And so hopefully you haven't forgotten all that, right? Because I made you learn it on purpose, because we're going to talk a lot about it. So that means that when ADH binds to the V2 receptor, it changes the shape of the receptor, which activates the G protein, right? So it'll exchange GDP for GTP, which will liberate the alpha subunit. The alpha subunit will travel across and activate the enzyme adenylate cyclase, which will then produce CAMP from ATP. So your CAMP concentration increases. CAMP always activates protein kinase A, all right? And protein kinase phosphorylates stuff, right? And in this example, we are going to phosphorylate the aquaporin protein directly, which is pretty cool. And you might ask, well, where are these aquaporins? They're sitting in vesicles, right? Just waiting to be inserted into the cell membrane. So if I want to increase a membrane-bound protein into a cell membrane, I produce a vesicle where it's inserted into the vesicle membrane, right? And so these cells are always producing a few vesicles that are filled with aquaporin, all right? Aquaporin 2 to be exact. And protein kinase A targets the aquaporins, and it actually will phosphorylate a serine group, if you really want to know, a serine amino acid on the aquaporin protein, which will light it up, right? So that dynein, which is a motor protein, if you remember, can bind now to the vesicle and walk it to the cell membrane. So as long as protein kinase A is active and I'm phosphorylating my membrane-bound vesicles that are heavy in aquaporin, then I can continue to insert aquaporin 2s into the apical membrane, and water will move from the filtrate through the cells and out into the blood. Now, the one thing I haven't told you is another aquaporin, aquaporin 1, is actually found on the basal side of the membrane, so that as water flows in, it can flow right out. It just flows right through the cell, right, into the blood, and the filtrate becomes really concentrated, right? Now, you might say, well, wait a minute, you might run out of these, right? I'm going to run out of vesicles. Well, protein kinase A also can be shuttled into, right, the nucleus, where it will phosphorylate the promoter, which is called a cyclic AMP receptor bound promoter here, or CRE. And I phosphorylate a Krebs protein in the nucleus, which can then bind to the promoter, and that will turn on aquaporin gene expression. What does that mean? That means I'll start transcribing the aquaporin gene in the nucleus of these epithelial cells, and I will produce lots of messenger RNAs that can then leave the nucleus, go to the ribosome, be translated into aquaporins. Those ribosomes will attach to the ER, producing vesicles, and then they will travel to the Golgi, be repackaged into the cell membrane, and then released so that you can increase the production of aquaporin vesicles. So protein kinase A has that effect. Now notice there's another enzyme here called phosphodiesterase. You see that? That breaks CAMP down, right? And so phosphodiesterases are working here, but as long as CAMP is being produced, because ADH is bound to it, it will overrun the phosphodiesterases. But once your blood osmolarity returns to normal and ADH stops being released, then we have to get rid of the CAMP, don't we? And we get rid of the CAMP using phosphodiesterases, so they break CAMP down. Isn't that cool? That's awesome, right? That's how that works. Questions on that? Again, what are you going to do? You're going to learn this. How are you going to learn it? You're going to walk through it, and then you're going to take out a whiteboard and draw it out for me, right? Or find a classroom and draw it out. I'm probably not in here because it's frickin' freezing, but that's right, the library. Now you know how ADH works. So what we're going to do, what's interesting is that in us mammals, ADH receptors are in the kidneys, in the collecting ducts, and the PCTs, or DCTs. I don't know why I keep saying PCT today, in the DCTs. Now, in an amphibian, the ADH receptors are in the skin, particularly the belly, okay? And so when you inject a frog with ADH, it's not going to work on the kidneys of the frog, it's going to work on the permeability of their skin. And they'll start taking water up through their bellies. And that'll allow them to swell or gain weight or to increase their blood volume, right? So when a frog becomes dehydrated, guess what? It doesn't get thirsty. It doesn't drink. It lays in water. So if you ever walk into my office and one of the frogs is sitting in its water dish, it's because it's dehydrated. So it's absorbing, it's drinking through its belly. And so that's one of the major behaviors of amphibians. ADH works on the bladder in reptiles, not in the kidneys, but in the bladder. And so the bladder is where you reabsorb water in a reptile. So it just depends on what vertebrate you're in. Kind of interesting, right? All right, as I mentioned, oops, why am I doing that? Sorry. As I mentioned, when you look at a nephron, particularly a juxtamedullary nephron, and you follow it, right? So here's my renal corpuscle, and I follow the corpuscle into the PCT, and then it drops into the descending limb, right? Makes a hairpin turn, goes up the ascending limb, and then when I go into the DCT, notice the DCT goes between the afferent and efferent arterioles. You see that? And so it's shoved between those two blood vessels and comes into really close contact with the glomerulus, with the afferent and efferent arterioles, right? And so if you were to take a cross section right here and to actually get a DCT in proper section, it would look like this. Here's your distal convoluted tubule coming at you, right? It's coming at you. It's between both blood vessels, afferent and efferent, and is basically sandwiched here where you have some cells outside of the two blood vessels. All right, and these cells have you can see they run like this way. See, there's some cells here and some cells here, and they're called JG cells, or juxtaglomerular cells. Juxta means next to the glomerulus, so you can see these cells in here. And then notice the distal convoluted tubule, the epithelium on the blood vessel side is enlarged. So there are columnar cells there, where the rest of the epithelium is cuboidal. And this structure is called the macula densa, one of my favorite structures. All right, this is actually one of my pictures. So from a mouse, here's a glomerulus right here. And these cells right here that are darker nuclei, that are a little more uniform, this is the macula densa. And you see the darker nuclei run underneath? Those are the juxtaglomerular cells. And there's a glomerulus. You can't see the blood vessels. I'm just not on the right plane. But you got a really nice image here of a macula densa. Here's another one up here of a mouse, again showing you a cross-section of everything. So here's the afferent arteriole, glomerulus, Bowman's space, right? Bowman's capsule, DCT, and notice the tall, darker cells here on the blood vessel side, macula densa. And then you see these little cells right here that are between the DCT and the renal corpuscle. Those are the juxtaglomerular cells. So a histologist is immediately like, whoa, this is weird, right? You got all these not, they're not coincidental, these things. You see it over and over and over again. So that allowed physiologists to start studying this setup. And if you think about it, it's in the perfect place, because these complexes together, the macula densa and the juxtaglomerular cells, it's called the juxtaglomerular apparatus together, is in a great place to monitor, isn't it? And you might be like, well, what do you mean? What do you mean, Dr. Gribbins? What do you mean? What I mean by that is the fluid leaving the DCT is the end product of tubular reabsorption and secretion, isn't it? Because it's what dumps into the collecting duct, isn't it? Right? And you're also really close to where filtration happens. So you're monitoring filtration and your end product at the same time, aren't you? Right? Isn't it? So you have the ability to do that. And these juxtaglomerular cells are really sensitive to things like blood pressure. They can sense the blood pressure of the afferent arteriole. Is it too high? Is it too low? They can also sense the filtrate, what the composition of the filtrate is. And that's what's the coolest to me, is they can actually know when there's too little sodium chloride or too much sodium chloride. They can metabolically sense, know when cells are hypoxically shocked or stressed. Do you guys know what hypoxia is? Have you heard that term before? Yeah, low oxygen, right? Hypoxia. So they can sense that, not by measuring oxygen, but by measuring byproducts of glucose metabolism. And you might be like, what? Why would, because you use oxygen to break down glucose, right? So let's talk about how the macula densa does that. So when the macula densa is stimulated, it will stimulate these juxtaglomerular cells to release, they call it a hormone, right? Renin. Renin really is an enzyme. It's an enzyme and a hormone, but it's really an enzyme in the way we're going to talk about it, because it is going to break down or cleave a protein that's circulating in the blood that has this name, angiotensinogen. So when you see ogen at the end of a macromolecule, it is an inactive enzyme. All right, that's what ogen means. And what renin does is cleave angiotensinogen and activate it into angiotensin 1. All right, angiotensin 1 will then circulate and will be activated by ACE. All right, the ACE receptor. You guys probably have heard of ACE receptor before because it's the site where COVID actually affects lung tissue. So it became really famous, right, during, well, if you paid attention to the science behind COVID. ACE is expressed in lung tissue. That's why COVID affected the lungs, at least the first couple of strains, affected the lungs more than any other body, right? Body organ, because its target is ACE. And what ACE is angiotensin converting enzyme. That's the name, angiotensin converting enzyme. And it converts angiotensin 1 into angiotensin 2. It's also a receptor. And that's why COVID spike protein actually binds to it. Isn't that cool? Right? And that's what they inject into you through the COVID vaccine. Do you guys get your COVID vaccines anymore? My wife makes me because it almost killed me, so. But a lot of people don't, right? But, you know, a lot of people aren't getting measles. They're giving their kids measles shots, too, because they're a bunch of freaking idiots. But, you know, then their kids die. And I told my wife, I said, those people, if their kids die from measles and they didn't get a vaccine, they should be, you know, they should be prosecuted for manslaughter. That's me, though. I'm pretty, that's just my opinion on it, right? In any case, this ACE protein here is what converts angiotensin 1 to angiotensin 2. And so what do I mean by that? Well, basically, and I don't have colored pens, but a lot of proteins that are released into the extracellular fluid are in an inactive form. And the easiest way to do that is real simple. You just add a few amino acids on the end of your polypeptide. And that will prevent the protein from folding up in the right three-dimensional shape. And so that's what angiotensinogen, it has a few amino acids on one end and a few extra amino acids on the other end. And when you cleave off those extra amino acids with renin, then one end folds upright and you get angiotensin 1. And then when you hit ACE, ACE cleaves off the other one because it recognizes angiotensin 1. And now the protein folds up into angiotensin 2. And this is a really active communication molecule. First and foremost, it is a potent vasoconstrictor. All right? So its immediate effect is vasoconstriction. And so vasoconstriction is when your blood vessels contract, and if you push on the blood, that's going to increase blood pressure, isn't it? All right? And so that's the immediate response of angiotensin 2, is to help increase blood pressure, which probably has dropped because of your decrease in blood volume, right? Or your increase in osmolarity. It also goes to the brain and causes the thirst response, like, damn, I'm thirsty. And you start drinking water, right? Or drinking something at least. You know, you know how a lot of people react to thirst here in the US? Is they eat. They don't, it's weird, man. They're not hydrated. And since they've never drank, they eat food, right? And so, you know, a lot of hunger, or what people think is hunger, is actually thirst. At least it's, you know, that's what some studies have suggested. And so if you drink a lot of water all the time, you can lose weight just by doing that, if people are having trouble with weight. So that, you know, whenever you do a weight loss program and you change your diet or you go into caloric deficiency, drinking fluids helps with the hunger response. Kind of cool, right? It also is going to stimulate vasopressin secretion. Didn't we just talk about that? So renin causes angiotensinogen 2 to be produced, which also causes the posterior pituitary to release ADH and concentrate urine. The other thing angiotensin 2 causes is the release of aldosterone from the adrenal cortex. All right, so you guys know where the adrenal glands are? They sit on top of your kidneys. They're two little structures that sit on top of your kidneys. And when angiotensin 2 reaches the adrenal glands, it causes the outer cells, or the cells closest to the surface, to release aldosterone. And then what aldosterone does is go to the collecting ducts and tell the collecting ducts to reabsorb sodium chloride. And remember, water follows solute, doesn't it? And so this will help not only absorb sodium chloride, but also help ADH absorb water out of the filtrate and back into the blood supply. Both will concentrate your urine. Now, what this little picture shows down here, and I know it's hard to see, is angiotensinogen. These are amino acids. Renin cuts off these guys right here. So renin, you can see it's shortened the protein to angiotensin 1. And then angiotensin 1 to angiotensin 2, you cut off just a few more amino acids right there, 2 of them actually, and now you get angiotensin 2. And so you're just cleaving the protein down into a smaller and smaller protein until it can take up its proper three-dimensional shape. All of these things, stimulus of vasoconstriction, thirst, vasopressin secretion, sodium chloride reabsorption, all will increase arterial blood pressure and blood volume, right? Make sense? Now the fun part. You're missing the fun part, girl. See, talking about all this thirst made her have to go to the bathroom. Oh, that was mean. All right, this is going to be I'm not going to, wait, you guys, have you looked at the slides? This is my favorite slide. Look at that one. Oh, that a boy, Angel. That's what I'm talking about. All right. Even if you're lying, you just made my heart grow a little bigger. All right, so how? So the question is, okay, this is cool, but how? How does the macula densa sense, right? How does the macula densa sense? So again, so this is kind of a drawing of this. So here is the afferent arteriole, right? Here are these cells that are sandwiched between the afferent arteriole and this over here would be the distal convoluted tubule. So notice you got a full epithelial cell here and then kind of a part of one there and a part of one there, right? So this is the basal surface of my epithelial cell of the DCT. This is my apical surface. And so this would be the filtrate right in here, right? This is a, let me turn the lights off here so you can see. This is a cool picture here. So this is a fluorescent microscopy. This is a confocal microscope, they're like $250,000, and they allow you to build kind of it almost looks kind of two-dimensional, right? And it shows you where the juxtamedullary cells are. So here are the juxtamedullary cells right here, these two green cells. Here's the macula densa, which doesn't stain here very well, right? But it's showing you the position of the glomerulus, macula densa here, and then the juxtaglomerular cells. Now, the juxtaglomerular cells are the ones that release renin. So these ones in green are the renin-releasing juxtamedullary cells. So the question is, how do we cause these cells to release renin? And how does the macula densa know when to cause them to release renin, right? Well, there's a little what they call paracrine molecule. This word is a good word to use because we're starting to get into the endocrine system, which is what we're going to do next. Anyway. So this molecule that we're going to talk about is a type of communication molecule that we call a paracrine ligand. Okay, so a paracrine ligand means local, a local ligand, a ligand produced that is going to act locally. So in this instance, it's going to work in the kidneys, right? Only, and it is called PGE2. All right, see it right there, PGE2, which is a prostaglandin. All right, so prostaglandins are paracrine signals. So it's prostaglandin E2. That's the name of what PGE2 stands for. There are lots of prostaglandins. Prostaglandins are often involved in things like immunity, inflammation, and other signaling pathways that are very localized types of signaling pathways. And what controls prostaglandin production is an enzyme called COX-2. All right? So you see it right here, COX-2. COX-2 is cyclooxygenase 2. All right, cyclooxygenase 2. And if you look at the notes underneath this slide and the really ugly slide, I list, the abbreviations of all these things. So you don't the ones I want you to know. Don't worry, you're not going to learn all this. All right, I'm going to show you a couple of pathways. That's pretty cool, though, right? Come directly out of a couple papers that have a review article that I found this figure on. It's awesomely awesome. I know, right? So COX-2, we're going to activate COX-2, and there's another enzyme here, notice, called prostaglandin E synthase. See that? These two enzymes, COX-2, cyclooxygenase 2, and PGES, work together to convert a fatty acid into a prostaglandin. So one of the more common fatty acids found in the cell membrane is arachidonic acid. And this is what arachidonic acid looks like. I don't care that you ever are able to draw it. Now, if you take biochemistry, you might have to draw it someday. But this is what it looks like. So notice it's a fatty acid that is unsaturated, right? It's got some double bonds on it. Now, what COX does, COX-2, is it converts it into PGE2. And this is what PGE2 does. So cyclooxygenase, it's a great name, cyclizes, see that? My fatty acid, and then adds oxygen to it. Right? That's what it did, cyclooxygenase 2, added oxygens and cyclized the arachidonic acid here and released it from my phospholipids to travel across to the renin cells. Now, you might ask how do we stimulate these two enzymes? There's a third enzyme called NOS, N-O-S, nitric oxide synthase. All right, nitric oxide synthase. Remember nitric oxide? It's a very important paracrine ligand, another one. And it's a gas, remember, and very hydrophobic. And so we're going to also activate NOS, which is going to produce nitric oxide, which will then travel across, enter the juxtaglomerular cells, or they're also called renin granular cells, and cause the release of renin. So how do we do this? How does the macula densa know to activate these enzymes? There's another group of enzymes here that are called ERK1 and 2 and P38. I just call these MAP kinases. Have you guys heard of MAP kinases before? So MAP kinase, which is a mitogenic kinase, it's one that activates mitosis, is also involved here, as you'll see in the next slide, which we're going to save for tomorrow, and I'll stop torturing you. But notice how we sense this. We can sense it through tubular salt sensing. Tubular salt sensing, so filtrate sensing, right? Now, remember, I told you the macula densa is much like the other parts of the nephron. It is continuously absorbing sodium chloride, right? The DCT does this, The collecting duct does it. The loop of Henle does it. So does the proximal convoluted tubule. But the DCT is no different. Notice what it has. It has a sodium, chloride, potassium co-transporter, doesn't it? See that? And we know what drives this, sodium potassium pumps, right? Which it's not on here, but there will be sodium potassium pumps over here, right? On the basal side. And you're pumping sodium out and bringing sodium in to move chlorides and potassium. We're most interested in sodium chloride, right? We also know that because we have a low sodium concentration in the cell, we can get rid of acids, right? And that's one of the major jobs of the DCT is to kind of, tweak pH, blood pH. So if we have a lot of acids, we can be pumping out hydrogens by using the sodium gradient. We can also do what's called metabolic sensing. And we do metabolic sensing, that is sensing if we're under metabolic stress, by monitoring succinates, all right? So monitoring succinate. So what the hell is succinate, right? If you guys have biochemistry, you probably haven't, have you? Guys, remember the Krebs cycle? What's the Krebs cycle? Don't worry, I'm not going to teach it to you. But what is it? What's the Krebs cycle? All right, where's the Krebs cycle happen? I don't remember. Where? Thank you, mitochondrial matrix, right? So the Krebs cycle is basically the finishing metabolic pathway to metabolizing glucose, right? So that we can collect protons and run the electron transport chain, right? Remember? Yes, Dr. Evans, I remember. Did I want to hear that again? No, but too bad, right? Now, if you go, and I'll let you do it, if you go out and just search up Krebs cycle, pull it up, an image of it, it'll be a cycle, right? And if you look at it, at the bottom of the cycle, right, where 6 is on a clock, there'll be a word succinate. Succinate is one of the intermediates of glucose metabolism. What drives the Krebs cycle and the electron transport chain is oxygen, isn't it? Right? So what do I mean by that? If oxygen levels drop, then everything backs up. The electron transport chain slows. NADHs aren't used anymore, right? And when NADHs aren't used anymore because they're not dropping off protons in the electron transport chain because of the lack of oxygen, then the Krebs cycle slows. And the first place that the Krebs cycle actually becomes hindered is at the succinate point on the cycle. Because I'm not going to, if you really want to know why, it's because it's the place where FAD is reduced to FADH2, which is the least energy step that in that breakdown of glucose, and is the first place where I will reverse what's happening because it's the more effective energy step. Or you can just trust me that you're going to end up stalling the Krebs cycle at succinate. And so what happens? You start building up succinate. And then what does succinate do? It leaves the mitochondria. And what does the cell do? Gets rid of it. Dumps it into the interstitial space, which it enters the blood. Ends up in the filtrate, where it doesn't belong. And if it gets to the DCT, that means you have lack of oxygen, right? Somewhere in the body because of a drop of blood volume probably, right? And dehydration. So that's what this S stands for right here, is succinate. There are succinate receptors on the DCT, and if you are having hypoxia or metabolic stress, then succinate's going to build up in your blood and end up in the filtrate where it doesn't belong, okay? All of these typically lead back to drops in blood pressure and blood volume. Cool, right? Now, if you have a drop in blood volume and a drop in blood pressure, then what's going to happen to filtration at the glomerulus? It's going to what? It's going to slow down. Good. Right? Your filtration power is going to slow down. And so what's going to happen to filtrate flow if blood pressure drops? That is, how fast is is it going to increase the speed of the filtrate, or is it going to decrease the speed of the filtrate through the nephron? Decrease, right? And so if you have a drop in blood pressure, that's going to affect afferent arterial blood pressure, which is going to slow filtration and slow filtrate's flow rate, right? And if the filtrate moves too slowly through the nephron, you're going to reabsorb too much sodium chloride, right? Because I told you the whole nephron reabsorbs sodium chloride, doesn't it? So if the flow rate's really slow, then the epithelium is spending more time in contact with the filtrate than it should, and it reabsorbs lots of sodium chloride. So when you get to the DCT, less sodium chloride's coming in than should come in, right? And because of that, this antiporter also slows, doesn't it? Because you don't have enough sodium chloride to pump hydrogens out. So the pH of this is going to change inside the cell, and the amount of sodium chloride coming into the cell is going to slow, and that's going to turn on these enzymes, COX-2 and NOS. All right, when you get a decrease of sodium chloride entering the DCT, or an increase in succinate, or a change in pH. That's how they sense. It senses this that way, right? The juxtaglomerular cells can also sense a drop in blood pressure because they're sitting against the wall of the afferent arteriole, and they can sense the heartbeat. And so if you get a drop, right, in blood pressure, they can sense that also. So it's like this strategically placed, these two groups of cells. All right, that's where